This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. Welcome to TWiM This Week in Microbiology. This is episode 153, recorded on May 25th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, how are you? Glad to be on.
I'm well. We're getting ready to go to a big meeting next week. Should be interesting.
Could it be microbe?
Yes, it could be microbe in New Orleans.
Where we're going to broadcast live?
We're going to... I don't know if
podcast live.
We're gonna record a TWiM there. That's for sure. I don't know if it's going to be live streamed or not. It might be.
Well, we'll have a live audience.
We will have a live audience, yeah. And that voice, of course, is from Ann Arbor, Michigan. Michele Swanson, welcome.
Hello. Sorry for jumping in before my introduction.
It's okay. We missed you last time, right?
Well, thank you. Yeah, I think so.
Okay. Well, welcome back, and also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you?
I'm doing well. It's been a busy week for me.
Yeah, well, here the semester's over and grades are in. I have to say, permit me to tell you that yesterday our oldest son graduated from college.
Oh, congratulations.
And he graduated with honors, too, which is great.
Oh, I'm not one bit surprised.
Well, he never listened to me. He is a cybersecurity major and will be working in New York City. He's got a job already lined up with a cybersecurity place, and so I'm excited for him.
Wow.
On his new life. Yep.
I need to hire him.
Yeah.
My youngest son just got a master's in social work from Wayne State here in Detroit.
Great.
Oh, wonderful.
So, and he's also got a very cool job that he's already started.
So we went to his graduation yesterday, Stevens Institute of Technology. They actually held it in the Meadowlands because they don't have a big enough place on campus. And I really enjoyed it because I love hearing the talks. I love the president. I love the guest speaker. My son hated it. He said, "Dad, why did you make me go to this?" But I said, look, all these good things that people said. And here's the best quote I want to take away from the meeting, which I think is really important from the graduation. The speaker said, "It's harder to be kind than to be clever." And his message was, just be fair and clever to people that you work, fair and kind to people that you work with. And my son didn't like that. He said, look at all these people in the world who do well and they're not very kind. And I said, yeah, but don't use them as guides.
And they could do twice as well if they were kind.
I think kindness and fairness, respect, these are all important things that we often lack, but I think they're so important. Anyway.
Listen, before we get started, shouldn't we mention that our colleague, Michael, has had a very lovely honor bestowed on him?
What did you have bestowed on you, Michael? Tell us.
I was awarded the Governor's Award for Excellence in Scientific Awareness.
Wonderful.
From the Governor of South Carolina yesterday.
You met the governor.
I met our governor. Yes, indeed.
Great.
He was very gracious, said many good things about science and the importance of science in our society and the importance of helping the public appreciate what science is all about. So it was a lovely ceremony, and I was impressed with how much time the governor gave to the awardees. In addition, there were 3 other individuals who won awards, one for excellence in research, another for a young investigator, and another for excellence in undergraduate education.
Michael, do you think that the governor listens to TWiM?
I don't know. I didn't have an opportunity to ask him. He was asking me about, I had written an editorial with the sitting governor of Arkansas, and it was in my package, and he was asking me how I met the sitting governor of Arkansas.
It all comes down to politics in the end.
Michael,
Access.
Congratulations. I mean, you're such a great role model, and we need a lot more people like you in these times.
You bet.
Well, thank you.
Yeah.
I want to tell you about this meeting that Michele alluded to. ASM Microbe 2017 is going to take place very soon, June 1st through 5th. That's a week from today in New Orleans, Louisiana, and this meeting will provide a unique platform to explore the full scope of the microbial sciences. It will feature an exciting program with opportunities for you to gain great new science and connections. And whether you're a researcher or an educator, a clinical or applied microbiologist, an infectious disease or environmental microbiology expert, explore the complete spectrum of microbiology at ASM Microbe 2017. For more information, go to asm.org/microbe. And if you listen to TWiM, you know the last few episodes, before the episode starts, there's an advertisement which you could use. There's a code in it, and you could get $50 off registration. And, if you haven't registered yet, it's probably kind of late, but check that out. Now, we're going to be doing an episode of TWiM at ASM Microbe. We're going to be doing it on Sunday, June 4th, and it will take place at 2:30 p.m. in the Exhibit and Poster Hall, ASM booth 1737. So come by and check us out. We have a guest who I'm sure you will enjoy. And it will be live streamed. So if you want to watch the live stream, 3:30 p.m. Eastern Time on June 4th, facebook.com/ASMFAN, one word, lowercase. 3:30 p.m. Eastern, facebook.com/ASMFAN. All right, we have today 2 papers for your enjoyment. We have a snippet and a regular paper, and Michael is going to start out with a snippet.
All right.
Time's up. Sorry.
Okay. First, this paper is posted on the BioR14 preprint server.
BioArchive. You say BioArchive.
Oh, is that how it goes?
Yep.
BioArchive. Okay, BioArchive preprint server. So it's in the public domain, so you can go off and look at it. It has not yet been subjected to peer review. We've all likely heard the phrase caveat emptor, or let the buyer beware. But today the phrase is caveat lector, and that's Latin for let the reader beware. Preprinting has been popular in the physical science world, where group science is the norm, rather than what we're mostly familiar with in microbiology, and that's investigator-initiated science that's so familiar to many of our listeners. I was delighted to see this paper, as I think it will help advance the field as they put their novel ideas out there to help the field progress. And I think these authors have every intention of publishing this paper in the normal way. And as we see more big science with genes and the microbiome, I think this sort of preprinting activity will become the norm rather than the exception. Paper was posted recently on May 8th. And it has been tweeted about 50 times, and the abstract viewed almost 1,000 times, and it's already been downloaded 200 times. So looking at the statistics, that's pretty good for a paper, even after it's been published. So there's a general interest in this particular paper. The topic of the paper brings together the concepts of type 6 secretion systems, the microbiome, and how the microbial players control who is admitted to this great country club that we refer to as the human microbiome. And to bring you up to speed, we did talk about type 6 secretion on a previous TWiM. I think Elio talked about it, and I'll get the number of the episode and put it into the show notes for folks. But type 6 secretion is a remarkable machine used by a wide range of gram-negative species to transport proteins from their interiors across their envelope into an adjacent target cell. And type 6 secretion systems have only been around or recognized for about the last 10 years, and they were first described out of Vibrio cholera, which, as many of you know, is a pathogen. So most people were looking at pathogens, but now folks are looking at type 6 secretion, and what they appreciate is that type 6 secretion mechanisms have been described in a quarter of all proteobacterial genomes. And as you can imagine, given that it was first described for a pathogen, what folks did is they went hunting, looking for what other pathogens use the system to make us sick. But today we're going to witness how this system is used, and now I'm going to steal the title here, the landscape of type 6 secretion systems across the human microbiome. The gut microbiome reveals its role in determining the community composition. In trying to understand how community structure occurs in our gut, and we all appreciate that the gut microbiome is this rich environment, how the communities form and how they are maintained is sort of done with a lot of hand waving. And so you can imagine that if a type 6 system is present, and what type 6 does is it injects an effector protein into another bacterium. And when that effector protein goes in, what it almost uniformly does is it interacts with the host protein of the recipient cell and shuts it down. And generally, this is an essential activity for that cell, and the cell is effectively dead. In addition to producing the effector protein, or the toxin, if you will, the gene cluster also produces an immunity protein to protect the donor organism as well as its friends that it wants to keep in its country club, or if I'm going to use also a metaphor of a gardener as it begins to trim the microbiome of this community. So now in trying to understand how these communities form, you can imagine that if a type 6 system is present in the coordinating species, and it can inject a toxic effector protein from a donor to a recipient that doesn't want, by simply bumping into that cell, you can then picture that it might be serving as the master gardener responsible for either picking the plants or at least coordinating how close they get to their neighbors. Thus, how some microbes might influence the community dynamic of the system that is our gut microbiome. And that's precisely what these authors set out to do. And the paper is fairly straightforward. It's divided into 4 sections, with the titles of their sections giving you an overview of their thinking. And their methods are those that we have talked about in the past, sequencing, and there's some tricks that they have done here. And the 4 titles of their sections are detection of the secretion pairs in the human gut microbiome and how they did things, the type 6 pairs and how they displayed low diversity within the human gut microbiome samples. And I'll expand on that as to why this low diversity of this type 6 secretion system is unusual. And then finally, they finish up with talking about how Bacteroides fragilis is important in developing the microbiome of our gut. The old-time microbiologists will know that B. frag, as it's affectionately referred to as by the anaerobic microbiologists in the audience, is the principal organism that we detect in our gut below the diaphragm. It's the big player. And so as they take us through the paper, they begin to teach us that the human gut microbiome is a dense ecosystem whose composition is paramount to its function. So they lay that out very nicely. Then they also introduce the second set of variables in forming the microbiome. Factors such as diet, immune status, and host genetics have each been implicated in shaping this gut community. But again, this is where they comment on the hand waving and where the bulk of their paper and their central hypothesis comes in. But it was not well characterized or understood that the contribution of direct interbacterial competition to the structure of this community, no one really has begun to decipher this. And that's what this type 6 secretion system, or their central hypothesis is the type 6 secretion system is coordinating some aspect of this community structure. So that's pretty much the bottom line up front. And then the rest of it is the meat of the paper of how they took us through this. And so in the meat of the paper, what they cleverly did is they sought to define the distribution of the type 6 secretion systems in the family of microbes common to the order Bacteroideales and explore its function in the human gut microbiome through several publicly available metagenomic data sets. So they didn't have to go out and get the metagenomic data sets. They effectively got them off the shelf. And so this is where we see the transition to big data. They're getting the data off the shelf, and they're fundamentally asking a question: what role does type 6 secretion play in coordinating the development of this community? So it's pretty neat of how they set this up. And so from their survey, they looked at these two metagenomic databases, and they asked, are there type 6 secretion systems present? And they learned that the answer was yes. And the human gut microbiome was replete with bacteria containing type 6 secretion systems. They then categorize them, and this is where they begin to instruct the naive audience members what the effector immunity factors are doing, and they refer to them as a genetic architecture or a GA group. And so for the sake of making my 15 minutes, they labeled these as group 1, 2, and 3. Now, groups 1 and 2 are often found in organisms, principally the Bacteroideales, that order, that are subject to conjugal transfer, while the GA group 3 organism is principally locked into the primary player in our gut, or the microbe Bacteroides flagellus. They then focused their investigation on the genes specific to the order Bacteroideales, and they observed a markedly low diversity of the type 6 genes in the human microbiome samples. Specifically, they talked about this antagonism, and that's what type 6 secretion is all about. It's this antagonism where one organism is injecting this effector molecule or toxin into another, and they don't have a specific immunity factor to it, and the net consequence is that they get displaced from the community. And so GA and G2 are the big players by themselves, but it's actually G3 that is restricted to B. frag, and this particular strain of bacterium is often unique to the individual. It effectively follows us through our lives. Their findings and other workers in this very elaborate English garden that is our gut microbiome, they're trying to understand the role that B. frag primarily plays in this antagonistic relationship, controlling who's there, how many are there, and they make the argument towards the end of their manuscript that it's primarily as a consequence of the selective pressure from closely related species. And
what does that mean? I read that. I didn't understand what it meant.
The other species, because they're living in close proximity and they're all taking nutrients from the same common pool, and they're principally degradative, and they're living in this complex anaerobic community where some of them are protein degraders, some of them are carbohydrate degraders, and living in the right proximity where someone's waste product, the waste fatty acid, is some other's carbon and energy source. And it gets into this conundrum there's what's emerging in the microbiome literature is this paradigm of acetate, propionate, and butyrate. And that combination of those short chain fatty acids, which are toxic and waste products of various microbes,
right,
are also involved in this coordination. So I think that's what is intended by the sentence, primarily faces selective pressure from closely related species.
Because they'd be more apt to be competing for the same resources.
Mm-hmm.
Their metabolic profile would be similar.
And so as you pointed out when I was pitching this paper to the team to discuss it today as a snippet, Michele brought up this is like the paper that we discussed in detail a few weeks back on public good. So that the microbiome is actually, and this is where we begin to make it almost, the microbiome has original thought, and it's actually looking out for the host. And but I think it's just this dynamic equilibrium. And so I think their study offers the reader an opportunity to think about the analytical framework for globally deciphering the forces that dictate the establishment of bacterial communities, and then more importantly, the maintenance. And that's the big holy grail of the microbiome research world, is trying to get good bacteria to live in our guts and keep us healthy and happy and not develop metabolic syndrome. That's it in a nutshell.
I wish him luck. I think this is extraordinarily complicated. Extraordinarily so, and I'm not very optimistic that we're going to have really global answers about what's going on very soon. Eventually, probably, but I think this is just dealing with thousands, hundreds, probably thousands of different species, all of which have their own quirks, and so this is almost a factorial situation. And I'm just not very optimistic that we're going to really understand more than we'll get a general view of it, which is what this is.
It's a general view.
In terms of really understanding the details, we got a long way to go.
It'll definitely take a combination of computational biology and then bench biology to test and learn what the simple rules are, and then to be able to integrate those rules into a more complex population, which I guess is what ecology is all about.
Yeah.
You know, the old-time ecologists are going to say to us, so what? We knew this. But I think the thing that I found intriguing about this is the type 6 secretion system, and the fact that there were only 3 principal players. And in their first detailed data section that I didn't have time to go in, they characterize these gene cassettes that make different effector molecules and different immunity molecules, and they're grouping them into this genetic architecture. And that was really pretty interesting to me, because they're taking the data and they're condensing them down to these little groups. And we have 3, which sort of gets to Elio's point about how do you get around the bigness of the numbers and, doing a controlled experiment. So
this is interesting. Type 6 is just going to be one, as Elio said. It's just one thing. There are going to be other modulators. And on top of it, this is just DNA sequence. I mean, we don't know that any of these are active, right? And
that is correct.
The assumption is, now, to take an overview like this, you need to look at the DNA sequence. But I hope at some point someone will take this as an inspiration and go in and say, okay, when we have this DNA for this type 6 here, is it actually working or not? Is it doing anything? And it's a lot harder to do that, obviously. But I'm a little unsatisfied that, the authors don't even say anything about this. They just assume that this is how it is, when in fact we don't even know that these DNA sequences are doing anything, right?
So if people are interested in this idea, how does type 6 secretion systems, these kind of aggressive spears that can kill off neighbors, how that can equip bacterial populations to segregate? I would send you back to TWiM 147 that Michael alluded to, that paper on Vibrio cholera or Vibrios and how they use the type 6 to afford to be able to secrete lots of enzymes that then generate nutrients. And so they've got to be able to ward off cheaters who just want to come in and feed off those nutrients. So anyway, just putting in a plug for that TWiM 147.
Yeah, there's also an earlier one, TWiM 54, in the paper we reviewed. They made movies of bacteria killing each other because they loaded them with fluorescent dyes, and you could see the dyes changing as the type 6 killing was going on. That was a pretty cool one as well. There are movies in that paper.
And remember, the type 6 machine apparatus is actually an upside-down tail.
That's right.
Of a phage.
So, yeah.
This paper has something for everyone.
So what do you all think about preprints and preprint servers?
Well, this is an interesting point. We did some bioarchive papers last year on TWiV, and we got some pushback from people who said, how can you talk about a paper that's not peer-reviewed? And my answer to that is, well, we are scientists, and we can read them and make a decision for ourselves whether it's in general a good paper, whether the conclusions are justified or not. And that's how I would look at it. If we got a bioarchive paper and we didn't think that conclusions were at all justified, we wouldn't talk about it. So, we can do our own little peer review here. And in fact, it might be an interesting exercise to see when this paper is actually published in a peer review journal, how different it actually is from what we see here. So a lot of authors have told me that they get a lot more interest when they put their paper up as a preprint. A lot more people see it initially. So I like it. I like the whole idea, as long as you realize the caveats.
So caveat lector.
What about the volume of literature and, just the practical problem as a reader? How do I make decisions about where I can devote my limited reading time?
Well, I think in your field, you're probably interested in reading what's going on. And so these are papers that will eventually be published. So I'm not sure, what fraction will go on to be published or not. So that's a good question.
So you wouldn't just scan table of contents of bioarchives. You'd want to be able to search for very specific terms that directly...
Depends. Like I would scan the whole table of contents because I would look for content for blogs and podcasts, but
sure.
I think most people who are running active labs would want to simply scan in their own fields, which you can certainly do.
Let me pipe in. I don't think the concept of preprints is bad. It used to be very popular in the very old days when everybody knew everybody and they exchanged preprints.
That's right.
Informally.
Yep.
Not as a publication, but informally. But look, my feeling is what the world needs is fewer papers, not more.
Yeah,
we have
I agree.
1 million papers published in Science a year. I mean, this is an absurd figure, and we just can't cope with it. So, yeah.
I went to Nature website the other day to look for a paper. I could not believe how many different journals they're publishing.
Right, just Nature alone.
But the number of journals is just
amazing,
rising exponentially. PLOS, all kinds, and Frontiers and everything else. I mean, enough.
Well, I mean, you're not going to stop it. It has to naturally come to an equilibrium,
right?
right?
I know, but science is not served by this proliferation.
So as the needle swings toward this very large volume of some preprints and some published works, do you think the pendulum will then swing back and society journals or certain brand journals will then become more important because it's a trusted, vetted source?
Well,
for our limited reading time.
Well, that's the history of publications. It's always been true.
Also, this new initiative by ASM, MicroNow, which is in beta, they're going to be picking bioarchive papers, and in fact, the ASM Journals is making it very easy for you to put a paper on bioarchive as you submit it to their journals, and many other journals are doing that as well. So I think this is here to stay, but I do think that there'll be a peak of interest, and then it will subside to some kind of average point. Yeah.
I think the case is easier to make from an author's standpoint. I'm just thinking about it from the user's standpoint.
Yes.
How do I walk into a new field and know during the limited hours I have to read where I should start?
And that's why I picked this paper.
That was my point.
It was on the new ASM website that's going to go live in a few weeks, and I was looking at what was on the front page. And they had this bioarchive paper, and this was the particular paper MicroNow was featuring.
Yeah,
and I said, okay, I'm going to go look at this because my professional society is commending this manuscript for my attention on its front page.
Okay.
So that's how I stumbled into this paper.
I think, Michele, the reader has to make their decisions of whether they want to look at bioarchive or not. I don't think you're going to lose anything by not looking at it. But for the authors, I think it's a good thing. For example, a few weeks ago on one of my podcasts, we had a group who had published a paper on ants. They made the first mutant ant, and they said they couldn't get it even reviewed at any of the big journals. Then they put it on bioarchive, and then suddenly one of the big journals contacted them and said, we're interested in having you submit this.
Right. Yeah. So again, an advantage to the author.
Exactly. Yes. So I think that's
to the
true.
reader, it's less obvious.
I agree. I think you have to make your own decision whether you want to look there or wait till it's published. And as I said, I don't think you lose by not looking at as a reader, right. I don't think you lose by not looking at the bioarchive. I could be wrong.
I just think we're going to look more and more then to a good housekeeping seal of approval to set priorities for our reading time.
Well, but you could, Michele, in a paper, at a preprint in your field, you could immediately look at it and say, this doesn't hold water. I'm not going to read this, right?
Sure. Yeah, but I don't know that somebody new coming into the field could do that.
No, probably not.
The other risk is the people that have big names, we'd flock to those papers first because they're a trusted source. But for the new investigator, it'd be harder.
Michael, did you mention the group that published?
I forgot because I was worried about the time. Yeah. Okay, this is Adrian Verster and Benjamin Ross, both sharing equal billing as first author. Matthew Rady, Yaquao Bao, Andrew Goodman, Joseph Magos, and Ilion Bornstein. And they're from the number of labs at the University of Washington in Seattle, as well as the Santa Fe Institute in Santa Fe, New Mexico.
And Yale.
And Yale. Oh, excuse me. Yeah, that's right.
Thank you, Michael. All right, I have a paper for you from PLOS Pathogens, and this is called Transient Microbiota Exposures Activate Dormant E. coli Infection in the Bladder and Drive Severe Outcomes of Recurrent Disease. We have two co-first authors, Nicole Gilbert and Valerie O'Brien, the PI Amanda Lewis from Washington University School of Medicine. And, in the PLOS journals, the co-first authors get a little yin-yang sign next to their names.
Yeah.
It's very cute. Very cute
nice.
And fun. All right, so this paper is all about urinary tract infections, and I want to remind you that was a subject on TWiM 114, Milestones in Blue. It's an episode we recorded out in Ann Arbor, and one of our participants, Harry Mobley, right, works on urinary tract infections. Millions of women have recurrent urinary tract infections, 1% worldwide, which is about 70 million. 25% of women who get acute urinary tract infections will experience a recurrence within 6 months, and it's often the same strain as the initial infections. And these are problems because they have to be treated with antibiotics, and this accounts for 9% of all antibiotic use. And also these infections can lead to life-threatening kidney and systemic disease, so the bacteria can go from the bladder up to the kidney and get into the circulation and spread elsewhere. So the dominant pathogen in these UTIs, as we call them, is E. coli. And what we're dealing with in this paper is the idea that the E. coli can establish reservoirs in the cells lining the bladder, and they can remain there. And then periodically, they can be reactivated to cause the symptoms of a bladder infection. And the question is, what triggers this reactivation? And there's some clinical evidence that women with bacterial vaginosis with overgrowth of Gardnerella vaginalis, these are at higher risks for recurrent urinary tract infections. Having sex is another risk factor for both bacterial vaginosis and urinary tract infection. So there's some idea that a bacterium that doesn't belong in either the vagina or the bladder is somehow triggering these E. coli urinary tract infections. So what they have here is a very cool mouse model where they show that exposure in the bladder to Gardnerella can drive the emergence of E. coli from bladder reservoirs. So again, Gardnerella vaginalis is a common member of the vaginal microbial community. Exposure can occur likely, for example, during sexual activity, and this could be a clinically plausible trigger of recurrent UTI. So the mouse model, basically they transurethral infect the mice with E. coli.
I was wondering, it looks like they're using human strains of Gardnerella.
That's right.
I wonder why. Doesn't the mouse have their own?
They might, but these do work.
That's right.
And
can't argue with that, but I would have thought,
yeah,
a mouse model is mouse bugs in a mouse.
Well, not always. With viruses, you can do mouse viruses in mice, or sometimes human viruses work, and then you tend to like to use that. So
I'm being picky.
That's okay.
Picky.
Plus, as we'll hear, they are able to then understand a mechanism that is not bacterium or microbe specific. So,
right.
So we can just think of this as a tool.
Yeah.
All right. So they inoculate these mice through the urethra with E. coli, and then they monitor E. coli in the urine. And they find that those that don't have any E. coli in the urine after about 4 weeks, this is about 80% of the mice, they're the ones that have latent reservoirs. The ones that continue to excrete, they get rid of those. They don't study them. All right. So now we have latently infected mice. You have E. coli in the bladder, but not in the urine. They then give these mice vaginal bacteria. And Gardnerella, it does not actually colonize the urinary tract. So it doesn't propagate inside the bladder, but it does trigger E. coli emergence into the urine. So you put Gardnerella into the bladder transurethral, and then you start to see E. coli coming back out. Whereas before, if you give PBS, you don't get the same effect. And they find that 2 exposures are better than 1 in terms of the percent of mice that are excreting E. coli. And these 2 exposures can be a week apart. And furthermore, if they look in the urine, they see neutrophils, which is characteristic of recurrent urinary tract infection.
They also put in heat-killed Gardnerella, and nothing happens. And then they put in a good bacterium, a commensal, and it also does not trigger the E. coli.
Crispatus.
Yeah.
Crispatus or Crispatus. Yes, that's right.
Lactobacillus. So it looks very specific.
It's specific for Gardnerella. That's
one.
right. And it's got to be living, right?
Yep.
Yeah. So that was my next one. L. Crispatus doesn't have the same effect, and that's recognized as a beneficial bacterium. Now, just in support of the idea that what the Gardnerella is doing is reactivating the E. coli, they take the bladder tissue and measure how much E. coli is in it. And after Gardnerella treatment, the amount of E. coli in the bladder is reduced. So that suggests that the bacteria are leaving the bladder reservoir. The next thing they do is ask, does Gardnerella cause exfoliation in the bladder? In other words, the epithelium lining the bladder lumen, do they come off? Exfoliation is a fancy word for.
And is that the mechanism for exposing the E. coli that's been sitting around in those
That's
cells?
right. That's exactly right. That's the idea. So they do scanning electron microscopy. They have gorgeous photos.
Aren't they though?
They're just beautiful, and some of them are colorized as well. And one is in the show notes. It's just beautiful. You see this lovely layer of epithelium, and then one cell here is coming off, basically.
And I was actually able to communicate with Nicole and Valerie, the 2 authors on this paper, and they said this was one of the most exciting days of the project when they had done the experiment, they had done the CFU data that you just described, and then they took it down to the microscope, and when they saw these beautiful images, they're like, wow, we've got a mechanism.
The cool thing here is that the Gardnerella causes exfoliation, whether or not there's E. coli in there.
Right.
It just this is an effect of Gardnerella being in the bladder, right? The cells come off, and if you put L. crispatus in, no exfoliation, and you can find these cells in the urine. You can find they're called urothelial cells in the urine, the cells lining the bladder. They come off, they go in the urine, and you can measure them, and they're more in the urine of Gardnerella treated than phosphate buffered saline exposed mice. So they think it's very likely that this exfoliation is part of the induction of recurrent urinary tract infections.
And in addition to these lovely scanning EMs, they provide us really clear cartoons in the figure so that you can visualize what they're imagining this all means.
Very nice, yeah. Now, it's known before that exfoliation is likely to involve programmed cell death, which we call apoptosis. So they look for some of the cardinal signs of apoptosis, which includes membrane blebbing, little bits of the membrane coming off and peeling off. And this they see in mice that are given Gardnerella with or without E. coli. There are 2 other markers of apoptosis. One is called tunnel, which is when a cell undergoes apoptosis, the DNA gets fragmented and you can label it in a certain way, and that is called a tunnel assay. So they see these cells are tunnel positive. Another measure of apoptosis.
Tunnel with one N.
Tunnel with one N. That's right. It's not the Holland Tunnel. Cleaved caspase 3 is an enzyme in the cell that is cleaved when apoptosis is starting, and they can also look for that, and they find it here. So basically, they see all the indicators of apoptosis. And if you look in women, this has been done in other studies, bladder biopsies from women with recurrent UTIs, you also see more apoptosis than in controls. The next thing they wanted to know is, would Gardnerella cause kidney infection? What happens in some cases, you get a bladder infection, but then the bad news is sometimes the bacteria can go up into the kidney and cause what we call pyelonephritis. And this is not good.
It gets serious, doesn't it?
Then it gets very serious. Yes, this can be life-threatening. So they wanted to know if Gardnerella has anything to do with that. And in fact, what they find is when they put Gardnerella in the bladder, they can detect it in 30 to 40% of the kidneys 3 hours after exposure, with or without E. coli. So it seems quite invasive, right? Just putting it in the bladder, it's moving up into the kidneys, and they find markers of inflammation in the kidney. So there are, again, cytokines in the kidney that are turned on during inflammation. There's a serum protein, creatinine, which is a marker of inflammation. They find all these markers.
Creatinine isn't a protein. Wait a minute.
It's a marker of kidney function.
It's a marker of kidney function. I did say it was a protein, but I misspoke. It says creatinine. It's a nice little molecule that is not a protein, but it's a marker of inflammation in the kidney. The point is, it gets into the serum, and you can measure it, and that's an indication of kidney inflammation. And they can treat these mice. Now, IL-1, interleukin-1, is one of these cytokines that's produced as a marker of inflammation. There's a receptor that IL-1 binds to. They can treat the mice with a antagonist of the receptor. So it will bind the receptor for IL-1 and block IL-1 from working, and that blocks kidney injury caused by Gardnerella. So Gardnerella itself can cause kidney injury and inflammation. And as you might guess, Gardnerella also makes the kidney more susceptible to severe E. coli infections. 6% of animals that are given Gardnerella and E. coli develop high titer infections and with abscesses in the kidney and splenomegaly, swelling of the spleen. This is not seen in mice that get only Gardnerella and very rare in mice with E. coli alone. They can isolate Gardnerella and E. coli in the spleen from the spleens of mice with abscessed kidneys. So in other words, not only can you have kidney pathology, but the bacteria can spread elsewhere, and in this case go to the spleen. And that's known to be a complication of these urinary tract infections. Now, in just some clinical data from women, 1% of healthy reproductive age women with cystitis, bladder infections, have pyelonephritis. And if they look to see in women with Gardnerella in their urine, the number goes up to 9%. So Gardnerella is a risk factor for pyelonephritis. And also in a clinical study, patients with Gardnerella in their urine are more likely to have a history of recurrent UTI and kidney infection than without Gardnerella. So clearly between the clinical results that have come before in this paper, Gardnerella seems to be a risk factor for urinary tract infections. And that's really the importance of this study. In a mouse model, it shows that transient exposure to Gardnerella can drive the emergence of E. coli from the bladder and cause development of urinary tract infections.
I really like the way they went from their mouse model and then pulled observations from the clinical literature, which are not all that well appreciated from what they've described. So making a link from the clinical data to really drive the next set of questions.
Yeah, they actually say that the finding of Gardnerella in the bladder and the kidney, which has been known before, has been ignored.
Yeah.
I think 25% of women with high urinary frequency or dysuria had Gardnerella in the urine. And they're really surprised, and they say, well, part of the problem may be that it's difficult to grow Gardnerella, so maybe the standard clinical microlab assays aren't picking it up. And
Right. But if you can come up now with a specific therapy to get rid of the bad bug, the Gardnerella Gardnerella.
Well, this is where I think the studies really kicks in. The use of the Lactobacillus crispatus is one of the relatively few cases where a probiotic is understood. We understand how it works. It displaces the Gardnerella.
Yeah, so there are data that suggests that doing intravaginal treatment with Lactobacillus crispatus reduces the incidence of recurrent UTI, for sure. It's been shown before. Now, the cool thing about that study is in that study, the authors said the L. crispatus must be displacing E. coli, but what they think in this paper from their data, they actually think it's displacing Gardnerella. And so now
you can displace E. coli when it's in intracellular.
Right.
Well, the E. coli is there. It doesn't get reactivated unless Gardnerella comes in. That's their idea.
Yeah, but I'm saying displacing the Lactobacillus can't displace the E. coli. That idea was not likely to be true.
No, I'm just saying whether or not that's what the previous authors said, and these
right.
guys said it's
before these data.
Yes, before. That's right. Of course, before. I mean, this is novel. This is the first time they say that there's a clinically plausible explanation for reactivation.
It's also interesting to think about this. In this particular paper, the reactivation, the trigger of the E. coli to escape from these intracellular reservoirs led to pathogenesis. Mm-hmm. But perhaps we could take this knowledge and design a therapeutic intervention to chase all the E. coli out of those reservoirs and then hit them with antibiotics to get rid of it for good so that women don't have these recurrent infections when they get extra Gardnerella in their urinary tract.
Do you think, though, that you're going to have enough of a persistent population? Because E. coli, I know, will form persister cells, and even a small fraction of the population at 0.3% will be refractory to antibiotics. You almost have to goose them to get them to go into their full growing state in order to get it to work. So I may be with Elio on this one, thinking giving them the lactobacilli in conjunction with your idea of getting them to come out of the woodwork, so to speak, may be a way of preventing the pathology. Because what you don't want to have happen is their beautiful model. If you look in the upper half of their model, that's what you don't want to have happen, is the abscesses in the kidney and the kidney damage and all of the other bad things that are happening as a consequence of that E. coli just swimming upstream.
Right. So it would have to be some combination therapy for sure.
I think part of the issue here is that when there's a lot of Gardnerella, that's because of dysbiosis in the vagina. So there's something leading to that because normally it's not there in high quantities. And so they suggest that for women who are at high risk for recurrent UTI, reducing their Gardnerella colonization could actually protect against even though they have E. coli in the bladder, it wouldn't be a problem if you don't have Gardnerella.
My question. I think this is a beautiful study, by the way. I do too. Like it a lot. But is Gardnerella the only likely?
No, who knows? They didn't look.
Pro-pathogen or pre-pathogen.
Maybe
not.
Others
in the bladder.
It's a good question. It's a very good question.
I like their term covert pathogenesis to convey that this is not just one molecule, one toxin triggering disease. It's this interaction.
Yes, covert. So the Gardnerella is often gone by the time, you have these E. coli infections. So it's not suspected, but it's the one that triggers it. Yeah.
And you don't see it.
Yep, that's right.
Let's hope it's as simple as it sounds. I mean,
is it ever?
It's never as simple as it sounds.
The key is they have to do some clinical trials and seeing simply if you reduce Gardnerella, is that enough or is it not enough, right?
Yeah.
But that's the beauty of having an animal model. You can say, okay, here is what we see. Now let's go into people and test it, right?
A simple clinical trial maybe is something as straightforward as women who enter a primary care practice for a suspect UTI simply collect and ask the question, are any of these women carriers of Gardnerella? And at the same time, when women go in for their routine physical each year, ask for a vaginal swab and ask the question, is Gardnerella there? And then your outcome would be whether or not that woman will develop a UTI more frequently than women absent the Gardnerella.
Well, they did say that there has been a study of that, and a quarter of women with urinary frequency or dysuria had Gardnerella in their urine. So, there's already a hint that there's Gardnerella when there is a problem with bladder infections.
And I want to emphasize they're doing this work at the Center for Reproductive Health Sciences at WashU Medical School. So they are perfectly situated to go back and forth between the clinical or the animal model with mice and then a clinical population.
I just saw, Michele, that...
Let me say a little bit of microbiology here also.
Go ahead.
Gardnerella is a very strange bug. It belongs to the bifidobacteria, which is, I always thought as being something like a lactobacillus, and they are in the actinobacteria. So they are they're supposed to be gram-positives, except that they're gram-variable. And this is very funny. So that it has a gram-positive cell wall chemically, but it doesn't stay gram-positive all the way, which is funny. It is also a facultative anaerobe. So there's a lot of microbiology here, which is a lot of fun. And if anybody's
interested in doing this for your journal club in your department, for example, I want to point out they have a beautiful illustration, final working model for the pathogenesis that is definitely worth looking at. It's a great teaching tool.
I just noticed, Michele, that Harry Mobley was the editor of this paper.
Yeah, I saw that too.
One last historical thing. Can I tell you, Gardnerella was named after Herman Gardner, an American bacteriologist who discovered it in 1955. So this is pretty recent stuff.
Yep.
Wow.
Yep.
I would have thought this is old bacteriology. It is not. So.
It's a real conceptual advance. And the work was done by, as we said, two co-first authors, Nicole Gilbert. She's currently an instructor in the Department of Obstetrics and Gynecology at WashU. She was an undergrad at Concordia University in Nebraska, where she studied biology and chemistry, and then did her PhD at SLU, as they say, St. Louis University in St. Louis, with Jennifer Lodge, where she studied Cryptococcus neoformans pathogenesis. And then her postdoc, of course, was here with Amanda Lewis. She recently got a K01 that is going to allow her to continue along this line of research at WashU. But she developed her interest in science early in life because her mom was a science teacher, and she credits her mother with her lifelong love of learning and exploring and her fantastic skills as a teacher. She has a fond memory of the annual Science Olympiad, and she refers to an egg drop contest. She then was able to go to the University of Nebraska Medical Center and do an undergraduate research experience through an REU program, and that really hooked her. As I said, one of the exciting days was the scanning EM, where they saw this exfoliation for the first time. Outside of the lab, she has had a really strong track record in publications throughout her career. But outside the lab, she is the mother of 3. Their ages 4, 6, and 8. She also teaches Sunday school. Their family likes to go out hiking in the state park and enjoy many of the activities like the zoo at St. Louis. She also has a leadership position in her community. She's currently the vice president of the board of directors of the Lutheran Development Group, which focuses on providing low-income housing and business opportunities for immigrants and refugees in St. Louis. She's also generous with her colleagues. She pointed out that Valerie O'Brien also was a co-contributor to this and deserves credit as well. Valerie is a TWiM fan. She's also currently finishing up her PhD in the Lewis Lab at WashU in the Molecular Microbiology and Microbial Pathogenesis program. She's on track to defend in July, and then she'll head to Seattle and do a postdoc with Nina Salama, who's studying pathogenesis of Helicobacter pylori at the Fred Hutch there. She was an undergrad at Wittenberg University, which is a small liberal arts school in Springfield, Ohio. She double majored in biochemistry and German. In fact, she went abroad for a year and took both chemistry and physics classes taught in German, and then stayed on and did a year of genetics research as a Fulbright scholar before coming back to WashU for her graduate work. She also tells the story that she had to get her appendix removed while she was in Germany. And while she was there, she had to be evacuated from her apartment building because one night construction workers discovered an unexploded World War II bomb in the foundation where they were working nearby.
Oh my God.
But and she, yeah. So all told, she wants to encourage any other undergraduates who have the opportunity to study abroad if you get the chance. She's also really thankful for the collaboration they have with Washington University's Center for Cellular Imaging, which allowed them to do this really beautiful scanning EM and X-ray microscopy for this project. And she also said she and Nicole really enjoyed the process of writing with their advisor, Manda Lewis. They sat in her office, for example, writing the discussion section. They each had a Google Doc open and would simultaneously edit as they were talking. So it was collaborating in real time, writing the discussion that really illuminates the conceptual advances of this paper so well. So that's the story of our two co-first authors.
Nice. Very good. Thank you, Michele.
Yep.
All right. That is TWiM 153. The next time you hear us will be from ASM Microbe in New Orleans, as I said. You can find TWiM at Apple Podcasts, at asm.org/TWiM. And of course, if you listen on your mobile device, which most people do nowadays, you have an application that you use to play it. There are many of those, and you can just find TWiM and subscribe to it and get every show as we release them. And we'd appreciate your support of TWiM and all the other podcasts we do. You can go to microbe.tv/contribute and find the ways you can help us out. Do send us your questions and comments. We didn't have any this week. TWiM at microbe.tv. Let's get going, folks. You're falling behind the other podcasts there.
They're holding out. They're waiting for a free book.
They're contest. They're waiting for a contest.
We'll start contests after Microbe. I do have a bunch of books to give away. We'll start them again. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. And as I said, we'll all be together, all 5 of us, at Microbe 2017 in New Orleans next week. If you want to watch the live stream, 3:30 p.m. Eastern Time on June 4th, facebook.com/ASMFAN, one word, lowercase. I want to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. Find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.